अस्सलाम
वालेकुम अस्सलाम बेटा। अच्छा जी, today we'll discuss transport of materials across a cell surface membrane. Then later we'll discuss cell signaling. So let's see. Transport across cell surface membrane.
सर आपकी वॉइस नहीं आ रही।
It was muted. So let's see. I'll review this again. This is our topic today, transport across cell surface membrane. We know there are chemical reactions going on inside the cell, in the cytoplasm of the cell. And for that, there are some nutrients or some substances needed in the cytoplasm. So they need to move from outside into the cell. And there are some products or waste substances produced inside the cell that need to be moved out of the cell. So for movement of these substances into and out of the cell, there are three main methods of transport materials across the cell membrane. One is a passive transport. Okay. Other is an active transport, and then we have bulk transport. The basic difference in active transport and passive transport is that the passive transport is without the use of energy, and active transport is with the use of energy. And in passive transport, molecules or substances move down the concentration gradient, whereas in active transport, they move against the concentration gradient. We will discuss what does down the concentration gradient and against the concentration gradient mean. There are three examples of passive transport: diffusion, facilitated diffusion, and osmosis. And there are two examples of bulk transport: endocytosis and exocytosis. We'll discuss them one by one. So let's see diffusion first. Okay. So what does diffusion mean? If we look at maybe a room, or maybe if you can take an example of a beaker. This beaker has water put into it. So this is water. And if we put some crystals of maybe some colored dye or maybe potassium permanganate, you will see initially the color will be darker in this region where the crystals are found. But gradually what will happen, this color will start, distributing to the entire of this water. And how does it happen? So molecules of water are in continuous random motion due to the kinetic energy, and same is with these molecules that we put into the water. The molecules of liquids, gases, and even solids, they possess some kinetic energy, and due to that kinetic energy, they show some one or the other kind of movement in them. In liquids and in gases, there are weaker intermolecular forces in between them. So consequently, there are more random movements of these molecules in liquids and gases. And due to these random motion or movement of these molecules, the molecules move from a region of their higher concentration to a region of their lower concentration. So if this is a region of higher concentration of these molecules of potassium permanganate, and this is a region where they are in a low concentration, so they would move from high concentration to low concentration. Similarly, if we take two jars, one is filled with carbon dioxide, and the other is filled with maybe hydrogen, for example, or maybe in oxygen, for example. So you see, this jar A has high concentration of carbon dioxide, and this has low concentration of no carbon dioxide in jar B. So consequently, what will happen? Carbon dioxide will move from A to region A to region B down this concentration gradient. And what happens to the hydrogen? Hydrogen concentration is higher in jar B and lower concentration in jar A. So if these two jars, you know, there is a partition removed from in between these two jars, the hydrogen will move from jar B to jar A until an equilibrium is established, until the concentration of hydrogen on both sides in jar A and jar B that becomes the same. And similarly, carbon dioxide will continue to move from jar A to jar B until the concentration on both sides that become constant. But however, if we look at this again, if this jar A and jar B are separated with a partition in between them, which allows the gases movement, from A to B or from B to E, B to A. And we have, for example, high concentration of carbon dioxide here in jar A, but yes, there is carbon dioxide present in B, but it is in low concentration, which we can represent these with bar charts. Okay, so carbon dioxide would be moving in both directions. Carbon dioxide will be moving from A to B, okay, and this carbon dioxide will also be moving from B to A. But there would be more movement because there is a high concentration here in jar A, so there would be more movement from jar A to jar B and less movement of these molecules from jar B to jar A. So this random mixing up of these molecules of carbon dioxide from A to B or from B to A, that is known as diffusion. So diffusion can easily be defined as the net movement of diffusion can be defined as the net movement of molecules of a substance from a region of their higher concentration to a region of their lower concentration. Okay, so this difference in the two regions where there is a high concentration on one side and low concentration on the other side is known as concentration gradient, concentration gradient, or maybe known as diffusion gradient. Okay, so for example, if we divide in this area into two regions, this is region A, for example, this has high concentration of some molecules, and this is region B, which has low concentration of the same molecules. So this is region A and this is region B. Okay, region A has high concentration and region B has lower concentration. So if there is no partition in between these two regions, the molecules will have random movement from A to B and from B to A. And there would be more movement of molecules from region A to region B. And this slope from A to B that we obtain, or the difference in between the concentration of these two regions, can be represented with this slope, and this slope is known as concentration gradient or diffusion gradient. So if the movement of the molecules is down this concentration gradient from region A to region B, then this is known as diffusion. And this movement is without the use of any energy. And this process continues on the concentration at the two regions that becomes the same, and that is what is known as an equilibrium is established. Okay, so let's see role of membrane, role of cell membrane. in diffusion. Cell membrane, we already know, is described on the basis of fluid mosaic model, in which a phospholipid bilayer has the phospholipid molecules arranged in such a way that their polar heads are outwards and their non-polar tails are facing each other. And this makes a phospholipid bilayer. Okay. So polar heads, you know, they are hydrophilic. These polar heads are hydrophilic. So they make hydrogen bonding with the water on their outer surface. If this is water present here, and if this is water present here, so they make hydrogen bonding. These polar heads, these are polar heads. They make hydrogen bonding with this water. Okay. And these tails are non-polar. These tails are non-polar. Non-polar tails. These tails provide hydrophobic barrier to what? To the polar substances and to the charged particles or to the larger particles. For example, glucose is a polar molecule. If this is a glucose, for example, so you know glucose is a polar molecule. It is water soluble. So this cannot, even if it is high concentration on outside and low concentration on inside, this will not cross through this phospholipid bilayer due to this hydrophobic barrier here. You know, this barrier would prevent this glucose moving through this phospholipid bilayer. Similarly, sodium ions will not move through, you know, this phospholipid bilayer as it provides a hydrophobic barrier. Similarly, the larger molecules cannot, you know, like starch, even if it is not water soluble and it is not polar, but starch, glycogen, and such larger molecules cannot, you know, cross through it. Starch, glycogen, these are larger molecules, so they cannot also cross through this phospholipid bilayer. However, non-polar molecules, okay, non-polar substances or non-polar molecules like gases, oxygen, carbon dioxide, they are non-polar. Again, they don't have any charge on them, so they can easily diffuse from high concentration, region of high concentration to low concentration through the spaces in between these phospholipid molecules. So oxygen can diffuse from high concentration if it is high concentration on outside, and this is low concentration on the inside. So they can easily diffuse through. This movement from high concentration to low concentration through the spaces in between the phospholipid molecules or the bilayer is known as diffusion. So this process is known as diffusion. Okay. Similarly, carbon dioxide can also diffuse out of it. Okay. So if this is carbon dioxide here, for example, it has high concentration here and carbon dioxide has low concentration here. So carbon dioxide will diffuse out, and this will be diffusion again. Ethanol, glycerol, steroid hormones, etc. These are non-polar substances, and they can cross, they can diffuse through the phospholipid bilayer. Okay. So that's why this cell surface membrane or cell membranes are partially permeable membranes. They allow certain molecules to pass through or certain substances to pass through them, and they do not allow the other substances to pass through. So non-polar substances can easily diffuse through the phospholipid bilayer, whereas the polar, water-soluble, charged particles and the larger particles cannot easily pass through this cell surface membrane. There are many other factors that affect, you know, the diffusion of the different molecules from a region of high concentration to the low concentration. So let's see rate of diffusion. rate of diffusion. The rate of diffusion can be calculated by using the living or the non-living materials. In non-living materials, we can use agar jelly to see the effect of different factors on the rate of diffusion. And in the living materials, we can use the plant materials like the potato strips or potato tubers to investigate the effect of different factors on the rate of diffusion. Factors that affect the rate of diffusion include, number one, factors affecting the rate of diffusion. It is temperature. The first factor is temperature. Okay. So temperature affects the kinetic energy of the molecules, and more is the temperature, higher will be the kinetic energy, and higher will be the random motion of the molecules of a substance, which speeds up the random mixing up of the molecules or the movement of molecules from their higher concentration to their lower concentration. For example, as I said earlier, if this jar half filled with water contains crystals of potassium permanganate into it, and this water is at low temperature, so this would take longer. This is at low temperature, so this would take longer for the potassium permanganate crystals to dissolve thoroughly in the water. But if we increase its temperature to the boiling temperature, this would increase the random motion of the molecules of water and that of the potassium permanganate molecules. So this would increase the rate of mixing of the potassium permanganate crystals into the water, and so they would dissolve more quickly or rapidly into the water. So this is boiling temperature, and in boiling temperature, these crystals will be very quickly dissolved thoroughly in entire of the water. Okay, so temperature does affect, or temperature increases actually the rate of diffusion. So more temperature, more kinetic energy, and more random mixing up of the molecules, and higher the rate of diffusion. Number two is steepness of the concentration gradient. Okay, so steeper the concentration gradient, faster will be the rate of diffusion. If, for example, we have this, a region A which has high concentration of a molecule of a substance, and this has, a low concentration of a molecule of a substance, and we can draw a slope from a region of high concentration to a region of low concentration. So that's, this slope shows the steepness of, the steepness of the concentration gradient. Okay, if the concentration at region B, so this is region A and this is region B, if the concentration of the same molecules at region B increases, then and even if the concentration of the A remains the same, but it increases after a while at region B, this steepness of the concentration gradient, that would become less steep. So this is less steep concentration gradient, and this is more steep concentration gradient. So if there is more steepness, the rate will be faster, and if there is less steepness, the rate will be slower. And the steepness of the concentration gradient is maintained by, for example, in the alveoli and the blood capillaries on their outside, by continuous flow of blood and by continuous breathing in and breathing out of the air into the alveoli. We'll discuss this gaseous exchange in detail in the lungs. Number three is nature of the molecules or particles. Polar molecules or polar substances have slow movement through the phospholipid bilayer, whereas the non-polar molecules have a faster movement through the phospholipid bilayer. Okay, then we have the size of the particles or the size of the molecules. Larger the size of particles, more energy needed to move them, more kinetic energy needed to move them. So slower the rate of diffusion, and vice versa. If there are smaller particles, they will have a higher rate of diffusion. Then we have surface area. More the surface area, more will be the surfaces for the molecules to cross through them, and so higher will be the rate of diffusion. And the last will be the distance or thickness of the exchange surfaces. Okay, shorter the distance, faster the rate of diffusion, and the longer the distance, slower will be the rate of diffusion. These are the different factors affecting the rate of diffusion. We will have some investigations on investigating the effect of these different factors on the rate of diffusion. You will have these investigations in your paper 3 in your practical classes, but we'll discuss their theory as well. Let's see some examples. Let's see if I could find a question on. Yes, this is illustrate the principle that surface area to volume ratio decreases with increasing size by calculating surface area and volume of simple 3D shapes, and then investigating the effect of surface area to volume ratio on the rate of diffusion. We will have some extended questions on this topic. So this is one factor. We will also have investigating the effect of changing surface area to volume ratio on diffusion using Agar blocks of the different sizes, and then obviously investigating the effect of temperature, and investigating the effect of even the different concentrations on the rate of diffusion. So let's see this simple concept of surface area to volume ratio. Let's see. Even you had this experiment today where you calculated the surface area and investigated the effect of surface area on the rate of movement of water. A student investigated the effect of changing the surface area to volume ratio on diffusion. Two different sized blocks of agar X and Y were made. The agar containing universal indicator solution. We have added some universal indicator solution into the agar. Agar is actually a colorless jelly-like substance that solidifies at a room temperature, and that is a dye mixed into it, and that makes an agar blocks that would have the color that the dye has been added into it. A universal indicator solution changes color when acid is added, and the blocks were placed in dilute hydrochloric acid at same temperature, and the student timed how long it took for each block to change color completely. Block X and Y are shown in figure 6.1. All dimensions are in centimeters. So this has, okay. So this X has this height, which is 2 centimeter, and this, the width, which is 1 centimeter, and this other dimension is 1 centimeter. So we can calculate its surface area, and we can also calculate its volume. Volume can simply be calculated by multiplying all the dimensions together, like 2 multiplied by 1 multiplied by 1. This would give us 2 cubic centimeters. So this is how volume can be calculated. Surface area, on the other hand, can be calculated. We can calculate the surface area of one side, we can calculate the surface area of this side. Okay, so this has one side 2 centimeter and other side 1 centimeter. Okay, so surface area of this side will be 2 into 1, and as there are two surfaces, one is this and the other is on the other side, so we will multiply it with whatever the answer we get. We can add that up together, or we can multiply it with 2. Okay, so that would give us 2 2s are 4 square centimeter. And then we have this surface. Even this surface has the same as we have calculated for it, and we have another surface here, so we can actually multiply it with 4 rather than multiplying it with 2, or we can multiply 2, 1 and 2 again, so that would give us 4 cubic square centimeter again. And then we have this side here and the side below, so it is 1 into 1 into 2, as there are two surfaces, so that would be 2. So we add them up together, so that would give us 10 square centimeter. So surface area will be 10 square centimeter, and the volume will be 2 cubic centimeter. And the surface area to volume ratio for this box will be, surface area to volume ratio will be 10 divided by 2, that would give us 5. So as this is a ratio, so this will not have any unit here. Similarly, we can calculate the surface area and volume of this larger agar block as well. So for this side and the other side, this side, the other, this second and this third and this fourth sides, they all have the same dimension. So this would be 6 into 3 into 4. So that would give us, you can make a calculation. And then we have this side and the side on the other lower end. That would be 3 into 3 into 2, and that would give us some value. We will add them up together, and that would give us the surface area of this larger agar block. And volume would be 6 into 3 into 3. So this would give us the volume. And take this value x from here and y from here, and we can calculate the surface area to volume ratio by dividing x with y, and that would give us some ratio here. So you will see, if you make a calculation, this value will be less than 5. Okay, so the point is, larger is an object, smaller will be its surface area to volume ratio, and vice versa. So smaller objects have greater surface area to volume ratio, whereas the larger objects have smaller surface area to volume ratio. Now let's see the question is the surface area to volume ratio of block X is 5:1 that we have already calculated. Calculate the surface area to volume ratio of block Y. Similarly, we can calculate the surface area to volume ratio for Y. Okay. These calculations are given here. 6 into 3 into 4 sides, 72, and you can do it yourself. Okay, so the ratio will be 90:54. You can simplify it, and that would give us less than 5:1. Okay, so we might have questions on outline. How would you determine the surface area of one side of a leaf? So you may be given a leaf as this is given here, and you have to find the surface area of one side of this leaf. How you will do that? There may be the different ways of doing it, but one of them is that you place a transparent, this is transparent grid which has a this divisions of some known dimensions on this acetate grid, a transparent paper or transparent grid, and this would be like this. And what we have to do is we know the size or the area of one box. We will have to count the number of boxes overlapping this outline of this leaf. So this is maybe 1, this is 2, this is 3, this is 4, this is 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. We can count the boxes that have a leaf covering more than half. Okay, and we can also include this. We can also include this, and we don't include this because this is less than half. So whatever the total number of grid is covering this leaf, number of boxes of grid covering the leaf into the area of one grid. So that would give us the total surface area of this one side of leaf. If the surface area of the both sides of the leaf has to be calculated, then we will multiply the surface area of one side with 2, and that would give us the surface area of the other side too. Okay, of the two sides. So that's how you should have done this. Maybe there are some other methods there, but that's very simple method. Let's see. Draw a round placed leaf on square paper or graph paper. Accept square ruler, count number of squares, and method of part squares. Add up all part squares and divide by 2. Match a larger part square with a smaller and count as one square. Or we have decided we will take more than half, and less than half will not be counted. Use graph paper with larger and smaller squares, or photocopy leaf, cut out and weight or divide by mass of 1 centimeter of same paper. Or reference to using an app, or further detail, place leaf flat on white surface, take photo single leaf. Though there is no mention of this method that I've described here, okay, the clear transparent grid is placed on it, and then the boxes covering the total area are counted and then multiplied with the area of one box. Though it has not been given there, but that's the correct method. You will get score for that. Okay. Now let's see. Organisms can be modeled as simple shapes for the calculation of surface area to volume ratio. Calculate the surface area to volume ratio of an animal modeled as a cube of side length 0.1 meter. Show your working. So if it is cube, then every side would be obviously 0.1, 0.1 and 0.1 meter. So if surface area has to be calculated, that would be 0.1 into 0.1. And as there are 6 surfaces, so you should multiply it with 6. And if volume has to be calculated, then it should be 0.1 into 0.1 into 0.1. And you can later calculate the surface area to volume ratio by, having this calculated here and this calculated here, and then surface area to volume ratio will be A over B. So that would give us a calculation here. Okay. And then we have an effect of changing surface area to volume ratio on the diffusion using the Egger block. As the surface area to volume ratio increases, the rate of diffusion will increase. And as the surface area to volume ratio decreases, the rate of diffusion will decrease. So you will have some investigations, and we will also discuss investigations in the lab as well. Okay. So that's it for today. We'll continue with the facilitated diffusion and osmosis in the next class, inshallah.
Allah Hafiz.
Allah Hafiz.